I will make fun of the entire time.
All right, everyone. I have a few announcements to start with today. So first of all, let's see. There is some concern that you guys, that a number of you raised earlier this week, that you were getting 0 out of 100 for certain media pages in the Achieve homework. Don't worry about that. It actually does not affect your score at all. I even went through, one person's scores and average everything up, and they don't affect anything. They're just the pages that actually only have written information trying to teach you some stuff, and there's nothing for you to enter. So don't worry about it. Hopefully you've seen the updates on the number of new tickets I've submitted to Achieve, including basically uncovering a raft of issues where they basically incorrectly use the rules for logarithms. And so many of you may not know these either. In fact, if I were to wager, most of you do not remember these rules at all, if you've ever learned them, right? So the rules for logarithms are, if you take a number with a certain amount of significant digits, and you take the logarithm of that number, the significant digits are supposed to be represented in the decimal points of the logarithm. So if you have two significant digits, 1 times 10 to the negative 7 1.0 times 10 to the negative 7 concentration of protons in a solution, right? And you take the logarithm of that, or the negative logarithm of that, to make pH. That should be a 7.00 is the correct number of significant digits. Because you had 2 significant digits for 1.0 times 10 to the negative 7, right? So you have 2 significant digits there. So those 2 are represented to the right of the decimal point in a logarithm. So the switch for the inverse is also appropriate. So if you're given a pH of 4.1, that's 1 significant digit. So that's what you would properly do if you were trying to determine what is your concentration of protons or OH minus or whatever, okay? Apparently, the people who programmed these new questions forgot that rule. And that is probably the main source of most of the issues you guys have been having, where significant digit rules just have not made any sense. And some of the things you've complained about, actually, they did them right, but I think it might have been largely accidental. Okay, so
it sounds like you're unraveling a conspiracy right now.
Yeah, right. So ultimately, I sent off a number of these new tickets and a very terse email explaining how this is not good, and ultimately, I don't think Macmillan can afford my hourly rate. And I've already put in a lot of hours on this. So I have lost patience. So any more of my patience that they're going to need to pay me for, or pay you guys for, okay? So so that's basically where we're at there. If you have any questions about those sig fig for log rules, I did put some of that information on some of the, on the discussion questions in response to some of the student questions, and also in that running tally of the different tickets. Okay. As far as I know, as of this morning, I hadn't gotten a response email that they had fixed all these things yet or evaluated all of them yet, but it's in the process. Okay. Please continue pointing out flaws. It gives me more ammo. Okay. All right. There are also, you might have noticed, for each of the different chapters, some quizzes and then the answers to those quizzes. Those are meant as study questions, right? So I really do recommend try to do those quizzes without looking at the answers first, and see if you get the right answers, or if you're off, why. And use the answer keys to help you figure out why you were off. And the reason I provide these is because everybody always wonders, what do your exams look like? Here's a hint. I make these quizzes to look exactly like my exams, in terms of sort of the types of questions, the structures of those questions, the types of things you should know. Okay, those are meant to be some level of guidance. They're not nearly comprehensive in terms of the types of things I could ask about, but they're to give you some idea of what to expect in terms of how the questions look on my types of midterms. Okay, so use them. They're a free tool. Okay. For those of you that have testing accommodations, now is the time to. Oh, go ahead.
I'm sorry. I forget if you told us this already or not, but are our exams going to be online or on paper?
They are going to be on paper in here. So back to the old way, right? The online was great because I could just sit in my living room and just like grade stuff on my computer. I'll actually have to have a stack of papers now, but you know that's why we're all back, right? So do it the old way. So for those of you that have testing accommodations, now is the appropriate time to schedule those with the Rowan Testing Services Center. Many of you haven't been on campus for the better part of 2 years and maybe have never been on this campus before. And so that you don't know that there's a Rowan Testing Services Center or where you can actually take tests. So it's very simple. After class, just Google Rowan Testing Center. And they basically have a way to request an appointment to take your exam. Okay, and generally speaking, accommodations, they usually have a line on there that testing arrangements need to be made a week in advance. So take that to heart. The testing center will fill up. So their appointments will fill up. So the sooner you book these things, the more likely you are to get something that will work with your schedule and not be like 9:30 p.m. at night or something, right? So generally speaking, try to schedule the test for the same time that everybody else would be taking it. A day early is doable. A few days later is doable. So October 6th is when the midterm is scheduled. 5, 6, 7, 8, those are all great days to try to schedule the exam if you want. Okay, but I would do that now. Okay. All right. Let's move on.
Dr. Fink, one
Yep.
more question. Is amino acids going to be the last topic that's going to be covered before the exam?
Protein folding.
Okay.
So we'll get there today. All right. When we left off, we were looking at this. Let's start shrinking and expanding things. When we left off, we were looking at this very short peptide, 5 amino acids. And you're all trying very hard to remember which amino acids these are, right? And ultimately, this is the type of thing where I would probably put a peptide or two on an exam and be like, man, which amino acids are these? So you should be ready to do that. Okay, just to give you sort of the basics on how we talk about peptides and build names for these things and understand these things. What is the standardized language for making peptides and proteins? We always start at the N terminus. Right, this is the N terminus, this is the C terminus, N for amino, C for carboxy. Peptides exist in aqueous solution, and so that's going to have a pKa, that's going to have a pKa. Generally speaking, these pKa's are similar to those of the free amino acids. So if I ask you, what's the approximate pKa of the amine in amino acid? Well, what's a good estimate? Yeah, if you said 9-ish, 10-ish, that works for me, right? How about the carboxy? 2-ish, yeah, right. So that means those numbers, right? Those are logarithms. And if your pKa is, say, 10-ish, that means it's 3-ish log units away from 7-ish, right? Which would be physiological pH, which means about 1 in 1,000 of these amines would be deprotonated or uncharged, and about 1,000 out of 1,001 would be protonated and thus positively charged, right? Henderson-Hasselbalch tells you that. Same deal with the carboxy. You'd expect the vast majority, this overwhelming supermajority of them would be in the deprotonated state and thus negatively charged, right? The side chains will each have their pKa's. The pKa's of the side chains are generally not affected too much by being in a peptide chain. They stay pretty steady, okay? And so you're going to memorize all these. You're going to know the vague details, right? Of these 5 amino acids, how many of them are protonatable or deprotonatable? 2? Right. Yeah, well, so you can look, right? Look for the nitrogens and look for oxygens, right? Generally speaking, right? So we've got hydroxyls here. This tyrosine hydroxyl group is has a pKa somewhere around 10. So it does occasionally deprotonate. And that's more relevant when it's in the active side of an enzyme. Right? What's this guy here?
As like the amino acids.
Yeah, the amino acid.
Oh, that's here.
Serine. Okay, good. All right, so does serine have a pKr? If you look back several slides?
No.
No? You know why?
Because it's uncharged.
Yeah, so basically it's unlikely to deprotonate in physiological pH, so why bother giving you a pKr of 15, right? So it does have a pKr. You can find a pKa for that, but physiological systems are not going to reach the pHs that would make that relevant. Okay, so that's why even though it has a deprotonatable group, we don't see a reported pKr for it. All right, when we name a peptide like this, we're going to go from the N terminus to the C terminus. So here's A1, A2, and so on. The way we will typically name them, let's see, okay, is not the way you could actually chemically name them. Why? Because it's a mess. If you can actually use the full amino acid names, and if you want to like have a fun game, I guess you could try to say 0-glycyltyrosyl-alanine leucine, put the marshmallows in your mouth while you do it and make it even more fun, right? That's a mess. It's hard to even figure out what's going on, right? So, and that's only 5 amino acids. We're talking about 15 in a peptide, or we're talking about a protein, that's not going to be any use, right? So what's far more typical is you're just going to use 3-letter or 1-letter codes, which is why you need to know them. So 3-letter and 1-letter codes will allow you to get all the information you need without having to try to smash them all together like that. Yes?
When we're memorizing the amino acids, does it have to be like we see the name and draw it, or just we see the molecule?
You should know the R groups. You should be able to draw any amino acid because it's just this core structure, and then you know the R group.
Is one of those naming conventions more common between the 3 letters and the 1 letter?
Yeah, so good question. So is one of these more common than the other? The shorter it is, the more common you're going to see like the full like 3-letter code. The longer, the more likely to see 1-letter code, right? It's really like a space and attention kind of detail. But they give you the exact same amount of information.
What are the nomenclature rules for the full amino acids?
For what?
The full amino acids, like is there like a nomenclature type of rule? Yeah, I'm just curious.
Yeah, like this is a perfectly cromulent way to write up the name of a peptide, however long you want that peptide to be. But like the returns are diminishing when it starts to take up more than 1 line on a page. All right. So this is why I make you learn those abbreviations and codes. Sometimes I'm just going to say, what is draw a titration curve for amino acid W. Who knows what amino acid W is? It's tryptophan. Yes. I always memorize it by picturing like an adorable 4-year-old trying to say it. It's tryptophan. So it'll also be really useful if you know how to draw peptides. So let's go back 1 slide here. This is one perfectly reasonable way to draw a peptide. Right? Notice all of the R groups are pointing up. and all of the oxygens and the amide bonds are pointing down, and all the NHs are pointing up in those peptide bonds. That's a perfectly reasonable way. This is clearly doesn't reflect any sort of geometric reality, right? But at this point, you know, if you actually tried to do stereochemistry here, you would drive yourself crazy, right? So this is perfectly fine. If I had to draw a peptide, this is a good way to do it. The other good way to do it is this way, where you can actually just like draw a squiggle as long as you need it, put the N on one end, then go 1, 2, and put another N, and then go 1, 2, put another N, and you basically have the backbone structure where the R groups are on alternating sides, right? And you can look at this and identify all those R groups, right? And notice this is also in a protonated and deprotonated form for the two termini, and also for the R group here. So ultimately, this is the other perfectly reasonable way to draw a peptide chain. And I give you this link to a website. I'm not going to pull it up on my pad here, but like it's a really useful website. Like you can add you know, amino acids, and it'll just click a button and it'll draw the peptide for you. But it also will do things like tell you the molecular weight and the overall net charge and what's the actual isoelectric point of the entire peptide. So the isoelectric point of an entire peptide would have to be the pH at which you get a net charge of 0, right? So just like you saw with individual free amino acids, if you have more than one pKa, you have to figure out what's the transition from -1 to 0 to +1, and then average those two pKa's, right? So you have a positive here, a positive here, and a -1. So you know you have 3 pKa's. So if I ask you to figure out roughly what's the isoelectric point of this, you'd have the same sort of logical exercise. You have to figure out, okay, as I go from more acidic to more basic, what is the charge going to be? And if you know roughly the pKa's of each of these, you know roughly what the isoelectric point of this little peptide would be. All right, so you make a peptide, you synthesize, maybe a short chain, maybe a longer chain, maybe a really long chain. It starts to fold into a protein. Okay. When we get into proteins versus peptides, again, I mentioned this before, but there's no like hard barrier where smaller than this is definitely a peptide and larger than this is definitely a protein. It's kind of a fuzzy boundary, and it really probably has to do mostly with what is the level of structure that this molecule will end up taking. Peptides are going to be less organized. Because they're smaller, they can only organize so much. And then proteins will be more organized. So the levels of organization have to do with the levels of folding. So the amino acid sequence, just the order of the amino acids, is what we call the primary structure. And sometimes that'll be a 1 with a degree sign of the primary structure of a protein. The secondary structure, or 2 with a degree sign, is the 3-dimensional arrangement of that peptide chain at the local level. And we're going to go through and talk about the main secondary structural features in just a little bit. If you have a large peptide chain getting into the protein realms, you probably end up with multiple secondary structures. And there's actually a sneaky one right in the middle here that we'll call super secondary. That has to do with how do these secondary structures locally interact with each other. And then the overall fold of the entire protein, so the 3-dimensional arrangement of all secondary structures, is the tertiary structure. So the tertiary structure is a single full protein completely folded. If that protein doesn't work all by itself, maybe it has other protein partners, then it can have what is known as a quaternary structure. The quaternary structure is the three-dimensional arrangement of multiple proteins. Sometimes those are called protein subunits, even though they're each individual proteins with their own genes and their own synthetic processes, but they're subunits of a larger mass that has its own job. All right, that process of multiple proteins coming together to form a larger, more complex structure is called oligomerization. So if I talk about oligomers in proteins, that's what that means. It is the conglomeration or the three-dimensional arrangement of multiple protein structures, making a larger, more complex structure. So you can visualize this a little bit here, right? Here's just the amino acid sequence, right? Here's just those three-letter codes that you all know and love now, right? Those three-letter codes have a ton of information, right? We are getting to the state where our understanding of the chemistry and the physiochemical properties of individual amino acids, and then what happens when we start to stick them together into peptides, is getting so good that we can just look at this sequence of information and predict reliably what kind of secondary structures are likely to form. This is an example of a secondary structure called an alpha helix. We're going to spend a lot of time explaining alpha helices pretty soon. And then this one secondary structure is shown right here, interacting with other secondary structures. You can see some other alpha helices here. There's some turns in there. There's no beta strands or sheets or something like that, but you'll understand what I mean by those soon enough. The overall fold of the entire mature protein is that tertiary structure. Now, this is an example of a protein that always, in its most active state, in its properly active state, has a quaternary structure. This is human hemoglobin. Human hemoglobin actually has two genes that produce the final mature protein. So there's what are known as alpha chains and beta chains. The alpha gene and the beta gene are themselves separate genes. They make separate proteins, but they're very, very similar. Their amino acid sequence are quite similar. There's a little bit of a tail. I can't remember. I think beta has a little longer tail. But they're kind of exchangeable. Like they're almost replaceable with each other. But what they do is they come together and form this larger larger structure where the two alphas and the two betas form a three-dimensional structure that with certain interfaces where they interact. And that'll be stuff that we'll talk about in a few weeks, about how those interactions between different proteins really, really matter for the overall function of hemoglobin. Okay? So in between here, I mentioned that there's super secondary, right? Super. Secondary. So that would be the arrangement of this particular alpha helix with maybe this one, right? Maybe there's a particular way that this alpha helix then loops into this next alpha helix and how they interact. Okay, we'll discuss that a little bit. It's a little kind of complex and kind of annoying that there's clearly this secondary and tertiary, and then there's enormously important thing in the middle that doesn't get its own like term within the number designation. As you look at the structure of human hemoglobin here, just note that you see patterns like this in nature, right? This, which is not it's not an accident, right? There are evolutionary relationships between species all throughout the more than the animal kingdom, to be honest, all throughout the domains of life that do similar things. What hemoglobin ultimately does is move oxygens around, like carry some oxygens. That's a useful thing if you have like internal aqueous environment where you need to move oxygen from the air to your cells, which is really hard to do just by absorbing oxygen through your skin. But it's also a useful thing to do if you're, say, small and you live in an aqueous environment, but you could use that oxygen for particular chemical reactions. So you see basically very similar types of proteins in cyanobacteria, this awesome animal called the gutless beardworm, right? And then giant earthworms, and that just because we're different and we have this evolutionary history, you can find sort of the same ways that nature solves a problem, right? But they may do it in really cool different ways, right? So, compared to the cyanobacterium, who can hold on to one oxygen and doesn't have any cooperative subunits, human hemoglobin has 4 cooperative subunits that can actually enhance the activity of hemoglobin. But look at the complexity of these different worms that are very distantly related from each other. They have massively larger hemoglobin proteins. So you always want to think about when you're trying to understand how evolution is working at this molecular level, we're not some sort of pinnacle of evolution. Everything that exists right now is a pinnacle of its own evolution, right? And over billions and billions of years, through trillions of generations or more, gutless beardworms and giant earthworms have solved.
Here's what I found.
What did Siri say about gutless beardworms?
I don't know, it didn't even pop up.
Gutless beardworms and giant earthworms and their ancestors have found upon molecular solutions for these same types of problems, some of which might even be far more efficient than our own hemoglobin, but they have a vastly divergent evolutionary history from us that we can only hope to learn from them. Up here at the top, it discusses a little bit about the types of symmetry. I know your textbook has some information about symmetry. I think that's only marginally interesting personally, so I don't really harp upon it in our lectures, and I don't really test on it. But it might be kind of interesting to you to sort of see how there's so many different ways that proteins that have complex subunit characteristics might interact with each other, and that symmetry is usually an indication of how the different subunits interact. Those sites, those interface sites between different subunits are often sites of particularly important activity. So when proteins form complexes, and they can be small, it could be just 2 proteins coming together. It could be ridiculously large things like there's 144 different proteins in this giant earthworm hemoglobin. And you're going to see some other complexes in this class that have hundreds of subunits. That is oligomerization, and that usually indicates that the functions of these proteins are incomplete without their partners. Okay? So the oligomerization state of a protein will affect its function because structure always dictates function. And you cannot get into these final structural shapes unless everybody's there. And then all of those modifications that we learned about to individual amino acids, those post-translational modifications, those affect final structural states too. They can affect how different proteins may interact, right? If you phosphorylate a serine or a tyrosine or a threonine, that can affect whether another protein subunit interacts with that surface of a protein. Other things that will affect the overall structures and thus the function of proteins include things like cofactors. Cofactors are a general class of usually freely dissolved, dispersible molecules. They sometimes be rather complex organic molecules, or sometimes they're just relatively small things like ATP. and sometimes even just little metal ions. Those are necessary for the function of a protein. If you lack a cofactor, your protein will lack its function. Heme groups we'll talk about, but that's an example just from the hemoglobins, right? That is a complex three-dimensional structure with a porphyrin ring coordinating a particular iron ion and keeping it at a particular redox state. Chromophores, similar idea. Some coordination of ions sometimes. Sometimes it's just a coordination of certain amino acid side chains that can produce colors. Lipids and sugars, all of these are required for the function of proteins. Now, that does not necessarily mean we know what the function or what its requirement is or what exactly it does. But generally speaking, if evolution has kept something around for a while, it probably does something useful. To give you some examples of how oligomerization can really matter in terms of structure and function, this is just a model of protein systems known as integrins. So integrins are transmembrane proteins, meaning they cross the lipid bilayer of the plasma membrane. So what you see up here is a little cartoon model where you have an alpha subunit and a beta subunit. And then this is the extracellular side, right? This is the intracellular space here. In order for integrins to actually have their function, the alpha and beta subunits have to interact and be activated by an internal factor. And when they do that, you might be able to guess from the name, they have a role of allowing certain cells to pass through different environments and allowing cells to integrate into those environments. So that's where the name integrin comes from. We'll actually give you an example of that using the immune system in a little bit. But in their inactive state, both subunits, the alpha and the beta subunits, do interact with each other. Right? And so what you see here, this is the meso squiggles. The red is one subunit, the blue is the other subunit. These are individual proteins, again, they're separate. And they have interactions up here, largely beta strands. You'll understand these arrows and squiggles a lot better soon. And then they also have interacting alpha helices through the transmembrane domains. And then inside, they have basically a site that can be regulated. And all it takes is an internal regulator to bind to one of those, pushing those two alpha helices apart. And then actually that ultimately extends the integrins out. You've activated the integrins. It's now basically a handle for other cells to potentially come along and grab onto this one. So a dramatic change in three-dimensional structure just from some organization down here, translated way out here. So this is
just pull apart.
Yeah, so like basically you'll disrupt the local interactions, basically kind of changing the fold and kind of rotating typically the transmembrane domains, so that they now no longer have things that play nicely pointing at each other. And so they spread apart, and that induces this extension. And now it's like you've extended out, but you've rolled out the red carpet for some cell to grab onto this domain up here that was hidden down here. So you can get massive changes in activity from relatively minor changes in one part of proteins. And then those activities are dependent upon the three-dimensional arrangement of multiple subunits. This is another example that I think is particularly relevant to college students. This is an enzyme, alcohol dehydrogenase. It gets its name because it is the enzyme that catalyzes the breakdown of alcohol in your liver, primarily. It is actually a dimer. One subunit alone has some coordinated cofactors here, including some zinc ions. But it won't work unless it has a matching partner. So two of these proteins come together and form what we call a homodimer or homomer. So it's the exact same protein, but they're flipped like 180 degrees from each other. And now you have this interface between them, which is the same face of both of the proteins. And that face, that interface actually is where most of the reactions occur. And so the two different subunits regulate each other, and they coordinate additional zinc ions and so on. And this catalyzes our breakdown of ethanol into acetaldehyde. It's actually the oxidation of ethanol to acetaldehyde, and then this is what clears ethanol out of your bloodstream on Friday nights.
Would the NADH in there be considered a super secondary or?
Oh, okay, good question. All right,
or
so
NAD?
so you see this NAD plus, NADH over here. NAD is a cofactor. Alcohol dehydrogenase does not work without those zincs either. Those are cofactors. So NAD is an electron carrier, and this is a redox reaction. And so you're oxidizing this ethanol into acetaldehyde. In the process, you are reducing NAD to NADH. This will all make so much more sense later on if that just sounded like gibberish. Okay, so they're cofactors. The secondary structures are each of these individual elements. The super secondary structures would be how, say, like this alpha helix is interacting with this beta sheet and so on. The tertiary structure would be the three-dimensional structure of one of these proteins. The quaternary structure is them together in their native or active form. The next chapter, we're going to deal with how do we fold all of those proteins. The end of this chapter is now, let's introduce some laboratory characteristics of working with proteins to you guys, because you're in the process of doing that, right? So you extracted M-cherry, which is a red fluorescent protein, last week. And this week, you're going to start some purification of that M-cherry. We try to plan these things, right? If we isolate proteins from cells, and they could be human cells, bacteria, and so on, right? They could be from any living creature. The first step is always clear out all the stuff you don't want, right? You got to get rid of the debris. Bacteria have cell walls, they have ribosomes, they have DNA, they have all sorts of things that you don't care about for the purposes of the lab you're doing right now. So how do we get rid of that stuff? Some of it's really easy, right? Just that initial spin where you pelleted out some junk, that's a purification technique, okay? So ultimately though, you're gonna end up with a mix of things that may have sort of similar characteristics, and it might be hard to separate them out. So better understanding how proteins get their physiochemical properties will help you better understand how you can start to purify them. So the process of purification, sometimes called fractionation, they will exploit the fact that different proteins will have different physiochemical properties based upon what is their amino acid sequence and what do they do, right? As you do that, you have to keep in mind, what is your ultimate goal, right? Do you just want to know what the amino acid sequence is of your protein? Do you just need a pure sample, you don't care if it works anymore? That means you can do one set of things. Do you need your protein to work? Right? Is it an enzyme that might be damaged if you use too harsh of methods? Okay, that limits the types of things you can do, right? So ultimately, if you're trying to study a protein's function, you probably have to be a little gentle as well. So we can take advantage of things like water solubility differences. Some proteins are going to be more soluble, some are going to be less soluble. Some naturally oligomerize with lots of different things, some don't, right? Proteins can be big or small, they can be positively or negatively charged or more or less polar, right? And they may bind to specific things. So some of the classic ways that you're going to purify proteins, you've already done, right? You've started that process, right? And others you'll never get a chance to do in biochem, but you may get a chance to do in the research labs you might work in, or at your job at post-row, right? So if we're making proteins for research, if it's really small, if it's just a little peptide, often we can just synthesize that in the lab using solid state methods. Okay? But those methods get less and less efficient the longer and longer the peptide is. And so things that are more than 30 and certainly more than 40 amino acids start to get really expensive to synthesize, because the purification processes necessary to make sure that you're only synthesizing and keeping the properly synthesized peptides get progressively more expensive. Okay, so if you want something that's longer or a full protein, right, we usually have to exploit biology to it. The easiest way is if you can trick bacteria or yeast or something like that into making it for you. So that's basically what we've done. What we took here, the right panel is basically the process that the bacteria in the biochem lab were tricked into making M-cherry red fluorescent protein. Basically, there were bacteria, E. coli, given plasmids, which is little circles of DNA that had, in this case, the gene for a green fluorescent protein, but just pretend it's red, right? And it's exactly the process here. Transformation is the process in which you insert foreign DNA into an organism. You transform bacteria, it goes inside, so you can see the little plasmids right here. That genetic information makes the protein production, the mRNA production and protein production machinery produce the necessary materials to create green fluorescent proteins within the bacteria, or red fluorescent proteins in the case of our lab. If you did this with mammalian cells or eukaryotic cells, that process is called transfection, but it's the same idea. You could grow them up, get a whole bunch of these bacteria, harvest them down, spin them down, make a nice little bacterial pellet, right? You remember this part now. Then break them apart. So how did you break them apart? Yeah, you sonicated them, right? So sonication, right? That is going to use basically the energy of sound waves to physically disrupt the lipid bilayers and the cell walls of those bacteria. What else did you use? Sorry, what?
Was it the buffer solution?
The buffer solution. What was special about that buffer solution? You're very quiet.
Was it the pH that I have?
It wasn't the pH. pH was should have been somewhere around 7.5, 7.4, and so therefore that wasn't causing.
We added salt.
You added salt, but there's salt in most things, so good idea. So it's not salt.
Is it hypotonic?
Yes. So you didn't add so much salt that it became isotonic. You kept it hypotonic. So if you recall in chapter 2, what happens when you put cells in a hypotonic solution? Where does the water go? Into the cell. So it goes from areas of high water concentration, i.e. low solute concentration, to areas of low water concentration or high solute concentration. Inside any cell, high solute concentration. So if water's rushing in and you're blasting it with sound waves, ultrasonic sound waves, right? That basically you're giving it a 1-2 punch and you're busting those bacteria wide open, and they spill their guts everywhere, which includes all the red fluorescent proteins and also all the things that you don't want, like the DNA and RNA and lipids and other proteins and so on, right? So now you got this mess that if you spin it down, right, we'll zoom in on purpose here. If you spin it down, right, you can pellet out a lot of that, right? So you did that spin and you got a bunch of junk in the bottom, right? And it was mostly not red because it was mostly not RFP, right? So now you can collect the supernatant and start doing other things. So when you guys do lab on tomorrow, you're going to do one sort of old school purification process where you're actually going to precipitate by deliberately adding some salts, and then spinning, and then deliberately adding salts again, and spinning, and so on. Okay. There are a variety of other methods that you could use. Many involve purification using chromatographic tools. So ultimately, once I get past this slide, we'll go into chromatography of proteins. I mentioned that if you were interested in relatively small peptides, that solid phase synthesis is probably your best way of making those at a high purification. Solid phase synthesis is represented in the diagram here on the right. There's a couple of interesting things to note when you're doing solid phase synthesis. Amino acids, no matter what their R group are, have a lot of reactive sites, right? And especially if they have a reactive R group, right? If you have an amine and you have a carboxylic acid for sure, and possibly an R group that is amenable to reactions as well. And so you're going to have to use things like protecting groups. So protecting groups, you add those to a specific site on a molecule. That makes them non-reactive. And then you can remove that protecting group later when you need it. It's basically like putting a cap on one thing, doing the reaction over here. Now you can take off the cap and you've got this new space that you can do the next part, right? And so with solid phase synthesis, you basically cap off the amine side because you don't want to react there. Glue the carboxylic acid side to a bead. Release the FMOC protecting group. Now you have that free amine, and it can react with a carboxylic acid of the next amino acid, and so on, right? And so this is a process in which you're going to add on the next amino acid, then remove the protecting group, and then add on the next amino acid, and so on. So it's a stepwise process. You'll have to basically kind of purify after the end of this reaction each time, right? You got to get rid of any unreacted free amino acid, and then mix in the next one, and so on, right? So it's a process that's going to have some efficiency, less than 100% each time, right? So if you can do the math, right? If it's 99% efficient each time, eventually you get to the point where the majority of the peptides you would synthesize will have an error in them. And so that's why the purification processes have to be like spot on, and that's why even though our technology has gotten really, really good, you kind of limited to like 40 amino acids, really. Okay, and then the other thing to notice is we actually synthesize these things backwards. So cells will synthesize proteins and peptides from the N terminus to the C terminus. We're actually synthesizing from the C terminus to the N terminus in solid phase synthesis. Okay, this is just a little image to explain what FMOC even is, and it just basically makes that amine less reactive. All right, so now to chromatographic methods of purification. This is the most common form of purification. It's actually often used in peptide synthesis to basically make sure that the peptides that you're working with are the ones that have the right length, size, and properties, and so on. It will allow you to separate molecules on the basis of their physiochemical properties, right? So we can use size, we can use polarity and charge, and we can use what do these things bind to. The general method for chromatography, or liquid chromatography, I should say, is that you have some sort of liquid or mobile phase, right? This is usually a buffer of some sort that your sample is going to be in, and you will add that sample to the top of a column, which is a stationary phase. So it's some sort of matrix of stuff, and depending on what you want to do, that matrix can be made up of sugars, it could be made up of silica beads, and all sorts of different things. But it's sitting there, and it has some physiochemical properties itself. and you're going to pass your sample through the column by basically pushing it along with the mobile phase. So the buffer is going to push it through. And as it pushes through, your samples are going to interact with the stationary phase. And then eventually it'll get to the bottom of the column and your samples will start to come out and they should be separated by their properties. Okay, so the simplest one is size exclusion chromatography. This is how you separate things by size, right? Ultimately, if you think about like a molecule moving through a matrix, the more things it interacts with, the slower it's going to move, right? Kind of bouncing along all the walls. So size exclusion chromatography uses porous particles and when building the matrix, so that there's basically little nooks and crannies. And those little nooks and crannies mean little things get stuck in the nooks and crannies, and big things don't. So they have less things to interact with. They kind of bounce through rapidly. So big things come out fast, and small things come out slowly. Okay, you can visualize it whether you prefer the left or the right, but the idea here is that small molecules like the little pink balls will get stuck in all the little pores, but big molecules like the red balls won't, and so they have less surface area to deal with, and they pass through more rapidly. And if you measure each of your samples coming out of the bottom, right? So if we look here, you know, you're collecting each of these samples here, right? If you measure, look at your samples, you should see a peak of the red beads far before you see the peak of the purple beads. Okay. Ion exchange chromatography is going to take advantage of the fact that some of your proteins are positively charged and some of them are negatively charged, and that the environment can shift that. Right? And if things are positively charged, they're more likely to stick to things that are negatively charged, and vice versa, right? So if you want to purify a protein that you think is positively charged, maybe because it has lots of lysines, right? Then you could use negatively charged beads in your solid phase. And negatively charged beads, positively charged proteins should kind of interact with them and stick to them, right? So you stick your mix of stuff on top of the phase, everything goes through, all the stuff that comes out early would also be likely negatively charged, right? Because they'll repel each other, and therefore they don't want to interact with the solid phase, and they'll wash right through. And then you'll have this resident population of things that are positively charged that are now stuck to the negatively charged beads. Okay, problem. How do you then get your positively charged protein off of the negatively charged beads?
Like wash it with a salt buffer, so that way you increase the salt and then the protein will get knocked over.
Perfect. If you didn't hear that, you wash it with a salt buffer. So you actually change the buffer characteristics that you start flowing through, so that you can change the charge distributions overall in the column. That's illustrated here. So what we have here, the y-axis is absorbance at 280 nanometers. You might remember that as the peak absorbance of tryptophan. This is the way you can determine basically roughly what is your protein concentration coming out in each of these fractions. The x-axis is fraction number. So basically, what's the number of the tube that's collecting the volume at this time, right? And so over time, fraction numbers will increase. What you see here is that over fractions 0 through 5, you get a big peak, right? This big peak is all these proteins that washed out right away. So they must not interact with the solid phase. They wash right through. Okay? Now you see a pattern of different other proteins eluting at different times. Right? So that's elution time is what that refers to. And you see this line going like this, right? That line refers to the right y-axis, the concentration of sodium chloride that is now in the buffer. As you add increasing amounts of sodium chloride, what is sodium chloride going to do? Well, the sodiums and the chlorides will disassociate from each other. And now sodiums, positively charged sodiums could interact with your negatively charged beads, right? And negatively charged chlorides could interact with your positively charged proteins, right? And so that will cause things to elute, basically at a concentration of sodium chloride that's going to basically displace your protein stationary phase interactions. So you would expect that this protein or set of proteins elutes earlier because it's less charged than these guys here, which took a lot, almost 1 molar sodium chloride in order to actually elute. So this is another way that you can separate things. So sometimes you'll do these things in tandem, right? Do that size exclusion first, but collect all the proteins that are likely in the 50 kilodalton range or whatever your protein is supposed to be. Now separate based on some charge characteristics. What else could you do? Well, if you know what your protein interacts with, you can exploit that. Is your protein an enzyme that acts on glucose? Well, if so, you could glue some glucose to your beads and then have your proteins just stick to that glucose, right? Everything else that doesn't stick to glucose just washes right through. But you take advantage of the fact that your protein interacts with glucose. Problem again, how do you get your proteins off the beads?
Can you add like an inhibitor?
You could try to compete it off, right? You know what's a good inhibitor? Glucose. Free glucose, right? If you just added some free glucose to your buffer, your proteins would exchange for the free glucose and just wash right through, right? So you could do several rounds of washing and then add some glucose, and that would elute off your proteins of interest. This is a good way to purify things that you design in the lab as well. Sometimes researchers will modify a protein's genetic structure so it produces a different amino acid structure to include a whole bunch of histidines, for example. You guys remember the structure of histidines. So histidines have those positively charged nitrogens depending on pH. Histidines are also really good at binding to zinc. And so you can have zinc affinity columns. So if you really, really want your protein and it has a whole bunch of these histidines all in a row, so it's really good at binding to zinc, that's how you can purify your specific protein that you want. So labs that try to get high purified samples of proteins that they've made up will often use things like that. You can do immunoaffinity too. You can have antibodies to your particular protein and use the ability of antibodies to have really high affinity binding to very specific targets as well.
Can you re-explain the free glucose thing with like inhibition?
So all you need, so the question was, could I re-explain the inhibition with the glucose example, right? All you need is something to compete with the beads, right? And so if you got glued on glucose, right, your proteins aren't permanently stuck. You have to think about this as an equilibrium. Your proteins will mostly be stuck, but they will come off and they'll find another glucose and stick again. So on net, most of your proteins are stuck at any given time. But if they're coming off and then they bind to a free glucose, and there's lots of free glucose around, now they're less and less likely to get stuck to the beads. Add enough free glucose, and almost all of the interactions are going to be with the free glucose rather than the beads. Does that make sense?
Yeah.
So I said zinc. I actually meant nickel. Sorry. But in any case, the idea is exploit the physiochemical properties of your protein, and then you can get relatively purified states of those proteins. And you guys are going to do that first with your ammonium sulfate precipitations, and then later on we're going to do some chromatography, and then we're going to run some gels and stuff too. If you're trying to purify something, you always want to make sure that you're only doing steps that enhance the purification, right? You're always going to have less than 100% efficiency. And sometimes the efficiency can be relatively low, but even if that efficiency is low, if you end up with a more concentrated sample of what you're trying to purify, that's probably good, right? So how would you make sure? Well, if your protein is red, you can just measure the amount of red per mass, right? So that's a way that you can measure the purification of your M cherry. Most proteins aren't red. It might be hard to find a particular wavelength that is particular to them, right? If it's an enzyme, you can actually measure enzyme activity. You can take a little bit of your sample each time and see it get more and more efficient at catalyzing a particular reaction. And that would be what we call specific activity. So ultimately, like if you do one round of purification, you should see a greater specific activity because you should get more enzyme activity per total mass of protein, right? 1 microgram of protein can catalyze X number of reactions. Purified, 1 microgram of protein should catalyze X times some number greater than 1 the next round, right? And so this should continually go up until you kind of hit whatever barriers there are to purification. There is a video quiz on polyacrylamide gel electrophoresis. So that's what PAGE is. Gel electrophoresis as a general tool is basically making a gel matrix of some sort and using that to separate your compounds of interest by electrical charge. So that's the electrophoresis part. Polyacrylamide is really useful for separating proteins. There are other matrices you might use if you wanted to separate DNA and RNA. We won't get into those really in class. But polyacrylamide gel electrophoresis, you make this gel matrix of acrylamide. You put your samples up at the top. You put a positive charge at the bottom and a negative charge up at the top, and proteins will migrate towards that positive charge. Why? Because you usually use some detergent that coats your proteins in a negative charge. So SDS is usually the detergent you'll use. So it'll unfold your protein, it'll coat it with a negative charge, they'll move towards that positive pole and spread things out. Now, it's not going to spread things out based on charge because your mass to charge ratio is actually going to be the same across all of your proteins. So it ends up separating things out by mass. And so big, fat, heavy proteins will stay up near the top of your gel, and small ones will move much faster through the gel and end up now down near the bottom. So it's a little bit like size exclusion chromatography, but it's upside down, right? The small stuff goes faster through the matrix. The large stuff stays up near the top. And this is a way that you can separate out your samples and figure out what it is you've even got. Once you've got your protein separated, you can stain your gel. So using basically simple Kumasi dye that turns proteins blue, you can see that we've got separated proteins out of different sizes. So here you've got a visualization of 2 different proteins, a much bigger one here, and it's coated in SDS. So now you have an unfolded negatively charged protein, but it's larger than this other unfolded negatively charged protein. The smaller protein moves faster through the matrix, so it ends up further down the gel. What you also see on the right with this example image of a gel is that there's a particular lane right here. We call this a ladder. And I have another slide specifically on ladders. Ladders are collections of proteins of known molecular weight, so known size, that you run alongside your samples so that when you get to this phase and you look, you can see, okay, these are the 14 kilodalton proteins, and these are the 29 and 43. You can see actually sort of a logarithmic separation between them. Yeah.
So I know you mentioned that there you have unfolded proteins. I know like with PCR every day, you do have the neutral, like the tertiary bonds, or secondary and tertiary bonds. Do you have to unfold them in order for this to work?
Yes.
Okay.
Because different proteins with, say, the same number of amino acids can have completely different 3-dimensional shapes, right? And the shapes would matter trying to go through a matrix, right? They could, some could be long and skinny, some could be squat and fat, some could be tighter, some looser, right? So if you unfold everything and coat it all in the same negative charge, it puts everybody on the same playing field. So,
What happens if you wanted to measure like the tertiary bond, like the tertiary secondary bonds?
so if you wanted then to, yes, so with this process, you would have to be unfolding the protein. So you would lose that tertiary structure for sure. You probably lose most of your secondary structures as well. Right? If you then chopped out one of these bands, so like scoop this out, right? You could, sometimes you can repurify the protein from the acrylamide matrix and then allow it to refold back into its native state and
Okay.
study it. This is not the best way to separate proteins and actually evaluate like the features of the three-dimensional features of a folded protein, because often this process is relatively harsh.
Yeah.
But it's a good way to evaluate how has your purification process gone.
Gotcha.
In this case, these are each different examples of proteins that you would find in, for example, just this first is non-fat dry milk, and then bovine colostrum. So colostrum is the milk that that mammals produce usually in the first week or two of the new baby's life. It's extra nutritious, so it has all the same things that you see in the non-fat dry milk, plus this extra large dose of immunoglobulins. So basically, moms donate antibodies to their babies. So what's useful here is all of these give you sort of some insight into what is collected inside dry milk or colostrum, right? So there's whey in there, there's casein, there's lactoferrin, all these different proteins and so on. So you can kind of identify like, okay, well, these bands are the ovalbumin, right? You can see them right here, basically. IgG is right here, and that's this, but you don't see that in non-fat dry milk. Yeah.
When we're purifying, what is like the contaminant we're trying to like take off?
So what are you trying to purify from?
Yeah.
Everything else. Like literally everything else, right? Like, so if you wanted to study, I don't know, let's say, if you wanted to study whey, right? Like, okay, whey makes up a small portion, or maybe that's actually these guys here. So whey makes up a small portion of the overall collection of stuff in milk, right? So you need to get rid of whatever this is, whatever this is, whatever this is down here. Right. And so that's basically the reality is you're getting rid of all the other proteins as well as all the non-protein stuff too. This is just a slide to show you the variety of different ladders that exist. Some of them are pre-colored for you, so you can actually see them while the gel is running. They can be completely different sizes, so you can change the characteristics of the gel itself. So maybe they're better at separating big proteins versus small proteins, and then you get a ladder to fit that as well. So we can use that polyacrylamide gel electrophoresis technique along with another technique, and we can create cool three-dimensional gels. So isoelectric points we learned about for individual amino acids, and you could feasibly estimate one just probably if you had the details of a particular peptide, you could probably estimate one. Well, a 50,000 kilodalton protein with 2,000 amino acids in it is also going to have an isoelectric point. There's going to be a pH at which its net charge is 0. And you can actually separate proteins by isoelectric point. You put them in a gel that has varying pH from one end to the other and apply an electric current, and the proteins will move from where you put them to the pH at which they have 0 net charge, because they'll lose their own motion. They will no longer be attracted to either the positive or the negative charge anymore. So you can separate by isoelectric point. You can take this column that you've made and actually plop it on top of a gel, just like we saw, instead of loading samples into the top of a gel like this, right? You put the whole column on top of the gel, and then you run it just like you would before. And so it will separate now by size. So you can separate proteins in 2 dimensions by their native charge, essentially, right? What is their isoelectric point, and then by size. And that can produce what we call 2D gels, where you have PI and size as the 2 separating features. And actually, here's an example of what those can look like. It looks like this because all these proteins were synthesized with a small amount of radioactive amino acids. And so then you can put this gel on top of an old school piece of film, and it'll actually cause the film to develop in just those little spots. And so each of these spots is mostly a different protein. I actually ran a few of these in my first job as an intern in a research lab, and it was hard. And you get a real mess like this, right? And ultimately, this mess, so here's another example. This is from a paper that just ran these 2D gels on a standard yeast strain, and then punched out each of these big spots and tried to figure out what the proteins were. So each of the numbers refers to the identified protein at that spot. So like number 221 here, they know specifically what protein makes that spot. And it's a relatively large protein, and it's relatively far to the right on this isoelectric point axis compared to everything else. Now, some of these dots are heavier and some of them are lighter. And that reflects that you have different amounts of different proteins at any given time, whether you're a yeast or a human, it depends on the cells, right? Some of them are darker, possibly because, for example, if you look at this one, there may be more than one state. Actually, you can kind of, oops. There may be more than one state of the protein. Maybe there's a dimerization that occurs, or maybe a change in the 3-dimensional structure of the protein causes a phosphorylation, for example, or something like that. Could make a heavier version versus a lighter version. Others could be really complicated because you can see some of these, clearly there's still overlapping dots, right? But kind of gets at the complexity of the types of, all right, come on. Kind of gets at the complexity of the different mixes of proteins you can have in cells. Just about done here. So when you finally get to a relatively pure sample of your protein, you need to know what the amino acid sequence is. If you know the gene, right, you can just look through the genetic code and figure out what the codons say, and that helps you, right? But sometimes we have the protein far before we have the gene, and we have to use the protein to go find the gene. So you need to know the amino acid sequence. This is not a trivial problem. You need to be able to basically identify each amino acid, not just the collection of amino acids, but where they are in order. Because if you don't have that order, you don't have the right information, right? So this was a massive problem for biochemistry. And it took one of the giants of biochemistry, Frederick Sanger, to actually solve this problem. Not a lot of people are double Nobel laureates, and this is a pretty deserving guy based upon the types of stuff he did as a freaking grad student. He developed this method. So anyways, he developed this method that would cleave off just the N-terminus end and allow him to identify that first amino acid. Now that wasn't enough, he couldn't repeat it. And so he would then chop up the protein and then determine all the N-terminus ends. And then he would chop them up in a different way and determine all the N-terminus ends. And then so on. And eventually he found the entire sequence of insulin. And that was the first protein sequence we ever had. Later methods, Edmund came along, figured out how he could repeat the process within sort of the same system and do it 50 times in a row. Now we have modern like mass spec techniques where we can blast apart a protein and just analyze the charge mass ratios of the different pieces and piece together the whole protein in a matter of a day. So those are different mechanisms to determine the amino acid sequence, and from that amino acid sequence, you get a lot of information. When we come back, we'll talk about the enzyme degradation. It's good to kind of just discuss this process, and then we will be able to hit the next chapter. All right, have a nice day, guys.
I don't have to go for it. I could walk that. Hey. Long story. I'll tell you that when we're walking. Have a nice weekend.
See ya.